So student, my screen is visible. Am I audible? So I'm launching poll, technical poll. You can answer through poll. Quickly answer me. My screen is visible. You can see my screen. My audio is clear. Okay. So today we are going to discuss cell, the unit of life, which is a very important chapter for NEET point of view. Okay. And today we are going to discuss the mind map of the chapter. Along with that, we are going to discuss some previous year questions so that you people can have an idea what exactly NEET questions are like. And you can also understand what are the important points on which you have to give more emphasis regarding NEET. All right. So this is a mind map of cell, the unit of life. As first thing is that cell was discovered by Robert Hooke, and that cell is the Koch cell. Okay, cell was discovered by Robert Hooke, and that cell is the from where he discovered that cell, from the Koch cell. Okay, and the first living cell was observed by Anton von Leeuwenhoek, and nucleus was discovered by Robert Brown. Okay, and Keith Porter discovered the endoplasmic reticulum. Your Camillo Golgi discovered Golgi apparatus. Similarly, George Pallet first observed ribosome. Chromatin was first observed by Fleming. Okay, these are the name of the scientist. So what type of question you can get in NEET is like they can ask you column A, name of the scientists are given in column A, and name of the discoveries or what they have observed in column B, and then the NEET can ask you like uh, match the following correctly, or they can also ask you a mismatch pair. Okay, this type of question you can get. Second one is the cell theory. As you know, that cell theory was first formulated by Sheldon and Swan. So Sheldon is the botanist and Swan is the zoologist. So Sheldon, what he observed the different type of plant cells and they form a tissue. All right. Different type of plants, he observed in plants the different type of cells, and all the cells they form the tissue. This was observed by Sheldon, and Swan worked on plant cell as well as on the animal cell. So it is the Swan which worked on both plant cell and animal cell. Remember this. Okay, in plant cell, Swan observed that the plant cell they have a unique character, and that character is this presence of cell wall. And in animal cell, they have a cell membrane, or we call it as plasma membrane. So Schwann proposed this hypothesis data that plants and animals, they're composed of cell and the product of cell. Okay, so Schwann, I'm giving this emphasis that he proposed the hypothesis that plants and animals, they are composed of cell and the product of cell. Okay, and Sheldon and Schwann together formulated the cell theory. But the cell theory is unable to explain that how the cells are formed, that how the cells are arising. This was explained by Rudolf Virchow. By the Rudolf Virchow, he proposed the omnicellular e-cellular, which means that all the cells are arise from the pre-existing cell. Okay, so the modern cell theory contains that all the living organisms are composed of cell and the product of cell. Second, that all the cells are arise from the pre-existing cell. Okay, and the cell is composed of cell are generally of two types. There's a prokaryotic cells and the eukaryotic cell. So the prokaryotic cell does not have a well-defined nucleus because there's an absence of a nuclear membrane. Similarly, the eukaryotic cell has a presence of nuclear membrane. So the eukaryotic cell has a well-defined nucleus. Okay, eukaryotic cell has a well-defined nucleus because there's a presence of nuclear membrane. In prokaryotic cell, they does not have a well-defined nucleus because there's an absence of nuclear membrane. Okay, all right, got this point? Next is the prokaryotic cells. Prokaryotic cells consist of your bacteria, which is a sole member, blue-green algae, mycoplasma, which is also known as PPLO. PPLO is like pneumonia, pleuropneumonia-like organism. They are also known as the joker of the cell. Because why? Because mycoplasma does not have a cell wall. Okay, so they can have a various shape. That is why they're also known as the joker of the cell. Bacteria, they are of 4 types on the basis of their shape. They can be a bacillus, which is a rod shape. They can be coccus, which is spherical. Vibrio is comma, and spirillum is spiral. Okay. And as I already told you that mycoplasma lacks cell wall. And these mycoplasma, they can survive without oxygen. And they also pathogenic in plants and animals. So and the prokaryotic, they have a genetic material, which is circular, single, closed, naked DNA. If I say naked, that means they lack histone protein. In eukaryotes, they have a histone protein. But in prokaryotes, there's no histone protein, so their DNA is naked. Okay? And apart from this genetic material or the chromosomal material, prokaryotes also have an extra chromosomal material. And that extra chromosomal material is known as plasmid. So plasmid is a small circular DNA. It is found in bacteria other than your genomic DNA. So plasmid ka function is to provide the extra I would say properties to the bacteria or a unique phenotypic characters to the bacteria, such as your antibiotic resistance to the bacteria. This you will get to know better when we going to study biotechnology, where the plasmid acts as a vector. So prokaryotic cell, they have a cell envelope, and the cell envelope is consist of 3 layers. Number 1 is the glycocalyx, which is the outer, and the glycocalyx is further divided into 2, which on the basis of their characteristics. If the glycocalyx is tough and thick and not easily washed off, then it is known as capsule. And the second one is loose sheath, which is your slime layer, because it can easily washed off. Apart from the glycocalyx, second layer is your cell wall. Okay, cell wall in case of bacteria in prokaryote is made up of peptidoglycan. Remember this, cell wall in bacteria is made up of peptidoglycan. You can write these points in your book, or you can write these points in your note or in your copy, because these points are very important. Okay. Next, from the cell wall is your cell membrane. And cell membrane of prokaryotes and eukaryotes are similar. That is phospholipid bilayer. Okay. And prokaryotes, as we know that in prokaryotes, the cell membrane bound organelles are absent. That means they don't have chloroplast, mitochondria, your endoplasmic reticulum, Golgi, lysosome, vacuole. They don't have the membrane bound organelles are absent in prokaryotes. Very important point, beta. Note this down. Okay. Then all the functions, like for example, the mitochondria function in prokaryotes is performed by mesosomes. So mesosomes, they are the extensions of the plasma membrane or the cell membranes. And these mesosomes, they can be of various shape. They can be a vesicle, they can be tubules, and they can be laminae in shape. And the function performed by these mesosomes, they are respiration, cell wall formation, DNA replication, and increase surface area of the cell or the enzyme content. So mesosome perform a very important function in respiration, because all the respiratory enzymes are present in mesosome. Okay, so the functions, so mesosome is analogous to mitochondria of eukaryotic cell. In prokaryotes, they have a flagella, they have pili, and they have fimbriae. So motility organ or the bacteria is able to move is because of the flagella, and the flagella is consist of 3 parts. One is your filament, second one is your hook, and the third one is your basal body. So the longest part is the filament, and the basal body is embedded in the cell wall and the cell membrane of the bacteria. Second one is your pili. Pili is beta, it is larger in size as compared to fimbriae, but smaller than flagella. And the pili, they are involved in conjugation. Okay, and the pili, they are also made up of a special protein which is known as pillin protein. Okay, this pili is made of pillin protein, and the fimbriae, they're the small bristle-like structure which are present on the bacteria, and they help in the attachment. They help in the bacteria to attach to the substratum. Okay, and in prokaryotes, the ribosomes are 70S. Okay, they are of 70S. The largest subunit is 50S, and the smallest subunit is 30S. Okay, and ribosome is the site of protein synthesis, right? And when there's a single mRNA and all the ribosome, like many ribosomes are attached to a single mRNA, that is known as a polysome or polyribosome. Okay, apart from all these characteristics, prokaryotes also have a one characteristic, that is the presence of inclusion bodies. Inclusion bodies are the bodies which are live freely in the cytoplasm. Along with that, inclusion bodies, they store reserved material. Okay, and they lack membrane. They does not have a membrane. Okay, and for example, like your phosphate granules, your siphonophysine granules, and gas vacuole. Gas vacuole provide buoyancy. It's a type of inclusion body which provide buoyancy in purple and green photosynthetic bacteria, and also in blue-green algae. I hope this mind map is clear to you all. You can answer me through chat, or you have any doubt regarding this. Okay, we are moving to the next one. So as I've already completed the prokaryotic cell, now we are going to discuss with the eukaryotic cell. Okay, so as I told you that the cell membrane is similar in prokaryotes and the eukaryote. They have a phospholipid bilayer. Though there are many hypotheses, or the model has been proposed regarding the cell membrane. There's a unit membrane, there's a unit membrane model, there's a sandwich model, but the most accepted and most important is a fluid mosaic model, which was proposed by Singer and Nicholson. Okay, so Singer and Nicholson said that the membrane is a quasi-fluid nature. Quasi means beta semi. Okay, that is a half fluid and half solid. Okay, it's a quasi-fluid nature cell membrane has, and the cell membrane, the majority, they consist of lipid, and mainly is phospholipid. The cell membrane is a phospholipid bilayer like this. This is your head, which is hydrophilic in nature or polar. This is your tail, which is hydrophobic and non-polar in nature. Cell membrane also have proteins. The proteins which are embedded or partially embedded or completely embedded, these are known as integral proteins. And the proteins which are present at the periphery, those proteins are known as peripheral proteins. Okay, it's very difficult to extract the integral protein because they're buried completely or partially in the membrane. So you can't extract the integral protein. But you can easily extract the peripheral protein because they're present on the surface. They're present on the periphery of the membrane. All right. Next, cell membrane also contains carbohydrate. You know about like glycoproteins are there in cell membrane. There glycolipids are there, right? Glyco is your word is for carbohydrates. Okay, remember this. When there's a glycoprotein, that means it's a protein plus carbohydrate. And the membranes also have cholesterol. So cholesterol is very important for maintaining the fluidity of the membrane. Okay, and what is fluidity? It is the ability to move within the membrane, that proteins can move within the membrane. So this is the fluidity, which is the ability to move within the membrane. And as you know that cell membrane is selectively permeable in nature. Okay, if I say selectively permeable, that means it does not allow all the molecules to come in, but it selectively allowed, like selective molecules can move in and move out of the cell. So the cell membrane is selectively permeable. Okay, remember this point. And the transport can be a passive as well as active. If I say passive, that means there's no requirement of energy. The movement of molecule is along the concentration. That means from the high concentration to the low concentration. And the passive movement can be of two types. It can be a simple diffusion or it can be a facilitated diffusion. If I say simple diffusion, that means it does not require energy. Molecules are moving along the concentration gradient, that is high concentration to low concentration, and there's no requirement of special protein. So in simple diffusion, there's no requirement of special proteins. Remember this point. Whereas in facilitative diffusion, because I'm using this term diffusion, that means there's no energy requirement. Along the concentration gradient, the molecule will move. And but in facilitated diffusion, special proteins are required. All right, special proteins are required in facilitated diffusion. Whereas an active diffusion, it requires energy. That means ATP is required. Against the concentration gradient, molecules are moving from low concentration to high concentration. So when we are making the molecule move against the concentration gradient, we requires energy, right? So this is an active process because it requires energy and molecules are moving against the concentration gradient. And the famous example is your sodium potassium pump, in which you required ATP to move the sodium and potassium in and out of the cell, right? Now the cell wall. As we know that cell wall is present in bacteria, as I told you, right? Cell wall in bacteria is made up of peptidoglycan, right? Fungi also have a cell wall which is made up of chitin, right? Algae also have a cell wall which is made up of cellulose, galactans, mannans, and minerals like calcium carbonate. Other plants or the higher plants, the cell wall is made up of cellulose, hemicellulose, protein, and pectin. Okay? And the higher plant, they also have middle lamella. Middle lamella, beta, it is the cementing layer which is present between the two adjacent cells, and this cementing layer help in joining the two adjacent cells together. Okay, and this cementing layer consists of calcium pactate that hold the cells together. Okay, I hope this is clear. And very important topic which I'm going to discuss is your endomembrane system. Is very important, beta. It's very, very important. Okay, so endomembrane system, there are four organelles which comes in endomembrane system, and all these four organelles, they show coordinated function. Okay, so these four organelles, they show coordinated function. Please remember this point. These four organelles show coordinated function and they comes under endomembrane system. And what are these four organelles? They are ER, which is your endoplasmic reticulum. Second one is your Golgi body. Third one is your lysosome, and fourth one is your vacuole, right? And all these organelles are single membrane bound. Okay, ER also have a single membrane, Golgi body also have a single membrane, lysosome also have single membrane, and vacuole also have single membrane, right? So first we are going to discuss endoplasmic reticulum. Okay, and remember this point, beta. Chloroplast and mitochondria. It is also given in your NCERT that chloroplast and mitochondria are not the part of endomembrane system. Remember this point, they are not the part of endomembrane system. Okay, it's very important. So first we are going to discuss endoplasmic reticulum. As I told you, the endoplasmic reticulum was first observed by Keith Porter. Okay, and endoplasmic reticulum, it has a two compartment. Okay, so inner side is your luminal and the outside of the ER is your extraluminal compartment. Okay, and the endoplasmic reticulum is of two types. Number one is your rough endoplasmic reticulum because it has ribosomes and it is involved in protein synthesis. Okay, it has a ribosome and it also has protein synthesis. It is involved in protein synthesis. And the second endoplasmic reticulum is your smooth endoplasmic reticulum. Why it is smooth? Because it is devoid of ribosome. Right? Ribosome nahi hai, because of that the surface is very smooth. They don't have ribosome, and smooth endoplasmic reticulum is involved in synthesis of lipid, synthesis of steroidal hormone, and also involves in the detoxification of drugs. Very important point. Write this down in your notebook, beta. Very important. Detoxification of drugs. Which organelle is involved? Is smooth endoplasmic reticulum. Okay. And the rough endoplasmic reticulum is involved in protein synthesis because it has ribosome. Okay. Now moving to the next organelle, which is your Golgi apparatus. It was first discovered by Camillo Golgi, right? It was first observed by Camillo Golgi. It consists of many cisterna. Remember, this consists of many cisterna. It has two faces, very important. Convex, which is also known as cis face, also known as forming face. Second one is your concave face, trans face, or the maturing face, right? So the vesicles which is coming from the ER or the endoplasmic reticulum is first received by your convex phase or your cis phase or the forming phase. So the first phase which received the vesicle from the ER, very important beta, it is received by cis phase. Okay. And after modification of the, whether it's a protein or a lipid, after modification of that component or after modification of the proteins or the lipids, these proteins and lipids, they moved out from the trans phase or the maturing phase or the concave phase. Okay. So Golgi perform beta two function. One is your packaging, and the second is the first one is the packaging of the material into the vesicles, and second one is the formation of glycoprotein or the glycolipids. If I'm saying the formation of glycoprotein and glycolipid, these are the modifications. So modification of lipids and protein, it taking place in the Golgi apparatus. Remember this. Now the third organelle which we going to discuss is your lysosome. So lysosome formed by the packaging in the Golgi apparatus, as I already told you, and they have hydrolytic enzyme. So lysosome contains all the hydrolytic enzyme, and these hydrolytic enzyme, they're active at acidic pH. Okay, so the enzyme will remain inactive till the condition is alkaline, and they become active once they get the acidic pH. Okay, these enzymes are lipases, the proteases, and the many more hydrolytic enzymes. I hope this is clear. If you have any doubts, let me know in the chat box. Next, Beta, we are dealing with the fourth organelle, which is your vacuole. So in plants, the majority of the portion of the cytoplasm is occupied by your vacuole, which is a 90% of the cell. Plant cell is occupied by vacuole. And the vacuole have a membrane which is known as tonoplast. As I told you that cell membrane is also selectively permeable in nature, right? Tonoplast is also selectively permeable in nature. Better remember this point. Tonoplast is also selectively permeable in nature, right? And these vacuoles, they contain water, sap, excretory products, and other waste material. So amoeba, they have contractile vacuole, which helps in osmoregulation and excretion. Osmoregulation is to maintain the osmotic balance of the cell. Okay, to maintain the level of the water, if the cell is requiring water so that end osmosis can take place, if the high amount of water is present in the cell, then extra water can be moved out of the cell. So osmoregulation or the osmotic balance of the cell is maintained by your contractile vacuole. Similarly, there's a food vacuole, which is formed by engulfing food. It is found in amoeba, right? So now we are going to discuss mitochondria and chloroplast. These are also very important organelle beta. Please pay attention to this, mitochondria and chloroplast. So first we are going to discuss mitochondria. As you all know that mitochondria, powerhouse of the cell. Why? Because ATP formation takes place in mitochondria. It is involved in ATP formation. It is known as the powerhouse of the cell. Important point, they are double membrane bound. They have an outer membrane and inner membrane, right? You can see this. This is your outer membrane and this is your inner membrane. The inner membrane forms of infolding, and that infolding is known as cristae. Okay, that infolding is known as cristae, where all your electron transport chain enzymes are present. Okay, and this fluid nature and this fluid component which is present inner to the inner membrane is known as matrix. Okay, and as you know that mitochondria and chloroplast, they both have a prokaryotic origin. Okay, they both have a prokaryotic origin. You must have studied about the endosymbiont theory. According to that, the mitochondria and chloroplast both have a prokaryotic origin, right? So if I say that they have a prokaryotic origin, there are following features which they have, which are similar to the prokaryote. Number one is your DNA, genetic material. In mitochondria, the genetic material is single, circular, double-stranded DNA, right? Which is similar to the prokaryote. And they also divide by fission. Okay, the bacteria also divide by fission. Similarly, mitochondria, they also have 70s ribosomes. In prokaryotes, the ribosome is 70s. Similarly, in mitochondria, the ribosomes are 70s. All right. Next is your chloroplast. So chloroplast, as you found in plants, it's a type of a plastid which consists of chlorophyll, and chlorophyll is a very important component regarding the photosynthesis, which is performed in chloroplast and in plants. So chloroplast, they have a membranous sac-like structure, which are thylakoids. And when these thylakoids, they are arranged in a stack, these are known as a granum. Okay, these are known as granum. And all these different grana, they are connected by a membranous tubule, and this is known as stroma lamellae. Okay, so they show a division of labor, because there's a thylakoid, there's stroma lamellae. Okay, so they show division of labor. And the chloroplast, they also have a prokaryotic origin. As I already told you, they have a prokaryotic origin. They have a ribosomes, which are 70s. They have a small circular DNA, similar to the prokaryotes, and the DNA is double-stranded. Okay. Now, next one is your ribosome. Okay, ribosome is involved in protein synthesis. So ribosomes are present in both prokaryotes and eukaryotes. In case of prokaryotes, the ribosomes are 70s type. In case of eukaryotes, in cytoplasm, the ribosomes are 80s. Okay, and this S represents the sedimentary coefficient and is also known as the Sedberg unit, because it was first discovered by the scientist, which is a Sedberg, who noticed this feature. So is known as sedimentary coefficient. It is the indirect measure of your density and size. So ribosome was first observed by a George Palade. The scientist's name is George Palade. Remember this. Ribosome is consist of RNA plus protein, and the membrane. They are membraneless. Okay, they does not have a membrane. Remember this point. And they are made up of RNA and proteins. Okay, I hope this is clear. Next is one here is the plasmid, right? Next is your plastid, sorry. So first thing is plastid contains chloroplast. So all these like chloroplast, chromoplast, leucoplast, they all are comprised in plastids. First, we are going to discuss chloroplast, which consists of chlorophyll and carotenoids. Second is the chromoplast, which consists of carotenoids and xanthophyll. Okay, and these chlorophyll, carotenoids, xanthophyll, they are fat soluble. Remember this, please write down this. Carotenoids, xanthophyll, chlorophyll, they all are fat soluble. Okay, and leucoplast is a colorless plastid. Remember this, leucoplast is a colorless plastid, and leucoplast further comprise of, one is amyloplast, which is store starch. Okay, you remember that starch is consist of two units. Number one is your amylose, and next second one is the amylopectin, right? This amylose, amylopectin. So whenever you get this term like amylo, remember this, starch. So amyloplast is a plastid that stores starch. Aleuroplast that store protein. Aleoplast that store oil. Okay. Next come to the cytoskeleton. Cytoskeleton is only present in eukaryotes. It is completely absent in prokaryotes. Remember this point. Write it down. Cytoskeleton present in eukaryotes. Okay. And cytoskeletons of three types. Majorly is microtubule, microfilament, and intermediate filament. Microtubule made up of tubulin protein. Microfilament made up of actin protein. Okay. And these cytoskeleton, they are proteinaceous filament structure, and they perform various functions like they provide support, they provide motility, they maintain the shape of the cell. Remember the spindle fibers in cells, you have studied in cell cycle and cell division. The spindle fiber are also made up of microtubule, right? The cilia and the flagella, they have exoneme, like 9+2 arrangement. That is also consist of microtubules, right? Then microbodies, beta, these are the single membrane bound vesicles. Okay, they are the minute vesicles, they are single membrane bound vesicles which contains various enzymes. Okay, and for example, like your peroxisome is a microbody, spherosome is a microbody. All right, you can see these microbodies in eukaryotic cell. So this is the last slide of the mind map, which discuss like cilia and flagella. As you know that cilia and flagella are present in prokaryotes, and the flagella is also present in, sorry beta, sorry. Okay, so we are going to discuss like cilia and flagella. So in case of prokaryotes, the flagella is consist of flagellin protein. Remember this. In prokaryotes, the flagella is consist of flagellin protein, right? In case of eukaryotes, the flagella, they have these 9+2 arrangement. They have this exoneme, which is the core, and they show 9+2 arrangement, where 9 is your peripheral microtubules, right? You can see these doublets. All right, you can see these doublets which are present at the periphery. So there are 9 peripheral microtubule doublets which are present at the periphery, and there's in the center you see a pair of singlet. Okay, you see a pair of singlet, and this one is your central sheath. So these 9 peripheral microtubule doublets are connected to the central sheath by the radial spoke. Okay, they are connecting to the central sheath by the radial spoke. Next, we are going to discuss centrosome and centriole. Okay, so centriole, you can see these. These are your centriole. They are made up of microtubules, and when the two centrioles are perpendicular to each other, along with the amorphous pericentriolar material, this whole structure is known as your centrosome. Okay, as you know, in cilia and flagella, the arrangement is 9+2. The exoneme has an arrangement of 9+2, where the 9 has a doublet, peripheral doublet, and +2 is your central singlet. But in case of centriole, the arrangement is 9+0. That means 0 means that there is no singlet in the center, and 9 means they have a periphery. At the periphery, microtubules are present, but these are in triplet, not in doublet. Okay, in cilia and flagella, they are present in a doublet, but in case of centriole, they are present in a triplet. Okay, they are present at the periphery and they're present in triplet. Centriole also forms the basal body of cilia and flagella. Okay, they form the basal body of cilia and flagella, and the central, they also form the they also consider like spindle fiber, right? They form the spindle apparatus, which is required for cell division. Most important we are going to discuss is the nucleus. As you know, that nucleus was first observed by Robert Brown, right? Robert Brown first observed the nucleus. It is a double membrane bound organelle, right? They have an outer membrane and they have an inner membrane, and these outer inner membrane, they are interrupted by these nuclear pore. Okay, and the space between the inner and the outer membrane is known as a perinuclear space. Okay, perinuclear space. As you know that all the cells, they contain nucleus, right? Nucleus is very important. All the cells contain nucleus, but there are few cells like RBC, sieve tube cells in plants, right? The RBCs and sieve tube cells, on maturity, they lack nucleus. On maturity, they lack nucleus. Remember this point. Okay, so nucleus, they have a like fluid component, and this component is known as nucleoplasm. So nucleoplasm is consist of nucleolus, Nucleolus, number one point, it is membraneless. Nucleolus does not have a membrane. Remember this point. You can make a chart and you can write all the organelles and whether they have a membrane, or if they are having a membrane, whether it's a double membrane or a single membrane. Please make a chart in your copy and you can write all these things because they are very important. Okay, so nucleolus is a membraneless and it is involved in the synthesis of rRNA, which is R is your ribosomal rRNA. Okay, it is ribosomal. Okay, so nucleolus is also known as ribosome factory. Remember this point, very important. Write this down. Nucleolus is also known as ribosome factory. Why? Because it is involved in the synthesis of ribosomal RNA. Okay, the nucleoplasm is also consist of chromatin. Okay, chromatin, which forms the chromosome. In cell cycle, you have studied that in interphase, the cell have a chromatin, like DNA is present, is loosely present and not in condensed form. That is a chromatin. But once the cell enter into the prophase, then the chromatin start condensing and it forms the chromosome. Okay. Chromatin have DNA, histone proteins, right? Now the chromosome, as I told you, that during the cell division or the M phase, you can see these chromosomes. So chromosome, they consist of chromatids, and the two chromatids are joined by the centromere. In the center, you can see these. This is a centromere. Let me know it. So this one is your centromere, and these are your chromatids. All right. These are the chromatid, and two chromatids are joined with the centromere. Centromere is also known as primary constriction. Okay. And this centromere, they have at both the sides, they have a disc-shaped structure, which is your kinetochore. Kinetochore is the point where the spindle fiber come and attached during your cell division. And on the basis of where this centromere is present in the chromosome, there are 4 types of chromosome are there. For example, if the centromere is present in the center and all the chromosome arm are equal, then it is known as metacentric. If the centromere is present slightly away from the center, then it is submetacentric. If the centromere is present towards the end, not at the end, beta, but slightly towards the end, then it is acrocentric. If the centromere is present at the terminal point, then it is your telocentric. Okay. Next, I'm going to discuss is your satellite. So satellite is the secondary constriction. You can see these. These are the secondary constriction. The satellite, they are non-coding. They consist of non-coding DNA. If I say the non-coding, that means they does not code for any protein. Remember these points. Okay, write it down in your book. That satellite, it consists of non-coding DNA. That means they does not code for any protein. Second is satellite. They show high degree of polymorphism. This you have studied during your DNA fingerprinting. There's a microsatellite, right? So these satellites, they show high degree of polymorphism, and these satellites, they also have repeated DNA. Okay, remember these points regarding satellite. I hope this is clear. If you have any doubt regarding mind map, please let me know, because we are going to proceed with the previous year question paper so that you can have more clarity about the concept. Along with that, people can know what type of question can ask in NEET. Okay, and you can also do a practice. If this mind map is clear, please let me know in the chat box so that we can proceed with the previous year question. All right, then we are proceeding with the question number 1. So question number 1 says, given below are the 2 statements. Okay, given below are the two statements. Two statements are given, beta. There's statement 1 and the statement 2. Statement 1 says, number 1 says that the membrane-bound organelles of the endomembrane system coordinate cellular function. Like membrane-bound organelles of the endomembrane system, they show coordinated functions. Statements 2 says that mitochondria and chloroplast are not considered a part of the endomembrane system. Okay, so you have to choose whether the statement 1 is incorrect and the 2 is correct. Second option says that both the statement 1 and 2 are correct. Option C, option 3 says that both statement 1 and 2 are incorrect. Or statement 4 says that statement 1 is correct, but statement 2 is incorrect. Okay, have you understood the question? Like, quickly answer me. Quickly answer, beta, in the chat box. Whether it's option 1, 2, 3, or 4. Like statement 2 are two statements are given. One says that the membrane-bound organelle of the endomembrane system coordinates cellular function, or mitochondria and chloroplasts are not considered a part of the endomembrane system, which is a statement 2. Quickly answer me. Payal, Sumed, Tivenshi, Joyce. Quickly answer. Okay. So I'm launching poll. If you want to answer through poll, you can answer. The poll has been launched. What do you think? Which statement is correct and which statement is incorrect? As I told you, the endomembrane system, it consists of 4 organelles. Remember those 4 organelles. What are those 4 organelles? Do you remember, beta? There's endoplasmic reticulum, there's Golgi apparatus, there's vacuole, there's lysosome. These are the 4 organelles which comes under endomembrane system. Okay, so let me help you out. Statement 1 says that membrane-bound organelle of the endomembrane system coordinates cellular function. As I told you, that endomembrane system is consist of ER, is consist of Golgi, is consist of lysosome and vacuole, right? All these 4 organelles comes under endomembrane system. Because in question they're asking the membrane-bound organelle of the endomembrane system. ER is also membrane-bound, Golgi is also membrane-bound, lysosome is also membrane-bound, and vacuole is also membrane-bound. Okay, and they all show coordinated function. Yes, it is true. I told you the important point of endomembrane system is that all the 4 organelles, they show coordinated function, right? ER, suppose this is your nucleus. Okay, I'm drawing it very roughly. This is your endoplasmic reticulum, right? And here comes your Golgi. This, okay, this one is your cis phase. This one is your trans phase. This one is your ER. And remember this, the outer membrane of the nucleus is continuous with the ER. Okay, outer membrane of the nucleus is continuous with the ER. Okay, so when the RER, suppose this is rough ER. Rough RER, it has ribosomes on it. Okay, I'm making the ribosomes. It contains ribosomes. So ribosomes are involved in protein synthesis. So maybe a vesicle. This is a vesicle formed from the ER and consists of protein. This vesicle will fuse with the Golgi, which phase? Cis phase, or we call it as forming phase. It fuses with the cis phase of the Golgi. Then the modification will take place. The modification of the protein will take place. And now this vesicle, which coming out from the trans phase of the Golgi, it contains the modified protein. Do you see that ER, Golgi, and this vesicle, if it contains, for example, if this contains a hydrolytic enzyme, then this vesicles can known as lysosome. Okay, so you can see that the function that these organelles are functioning in a very coordinated fashion. Like first this is ER, then Golgi, from the Golgi, then lysosome. Okay, they are arranged in a very coordinated fashion and they're working coordinatedly. Okay, so first statement is absolutely correct, right? Now, statement 2 says that mitochondria and chloroplast are not considered a part of endomembrane system. Like mitochondria and chloroplast. As I told you when I was discussing the mind map, that mitochondria and chloroplast, they are not the part of endomembrane system, beta, because they does not show this coordinated function. You can see that chloroplast is present in plants only, right? And the function of chloroplast and mitochondria is not coordinated. So both the statements are correct, whether statement 1 or statement 2. Both the statements are correct. I hope this is clear. So the correct option is option number 2. I hope this is clear. Let me know in the chat box. Payal, Sumedh, it is clear? Okay, all right. So we are moving with the next question. So question number 2 says, which of the following pair of organelle does not contain DNA? Which of the following pair of organelle does not contain DNA? So option 1 says mitochondrial lysosome. Option 2 says chloroplast and vacuole. Option 3 says lysosome and vacuole. And option 4 says nuclear envelope and mitochondria. So let me know in the chat box what you think is the correct option for this question number 2. Please, student, try to answer. Okay. One says C. Okay. Anyone? Try, everybody, please try to answer. Okay, Pyle says option C. What about others? At least try to answer better. Then only you will able to understand the question. Joyce, Sumedh. What do you think? What will... Okay. One says is B. Okay. Any more? More guesses? Sumedh, what do you think, beta? What would be the correct answer for this question? Okay, so let me help you with this question. It says, which of the following pair of organs does not contain DNA? Remember, they're asking about does not contain DNA, right? As I told you that mitochondria and chloroplast, they both have DNA, okay? They both contain the genetic material. They have their own genetic material. I told you that they have a prokaryotic origin. Okay, they have a prokaryotic origin. So their genetic material is similar to prokaryote, which is single, double-stranded, circular DNA. Okay, so let's come to option number 1, which says mitochondria and lysosome. Beta, mitochondria. Okay. So, let me just read out the question again. Which of the following pair of organelles does not contain DNA? Okay, does not. So, as I told you that mitochondria contain DNA, right? So, mitochondria contain DNA, but lysosome does not contain DNA. So, but mitochondria is there. So, this option is wrong. Option 2 says chloroplast and vacuole. Beta, chloroplast contains DNA, right? But vacuole does not. So this is wrong. Third one is your lysosome and vacuole. Lysosome and vacuole, which is your correct option, that lysosomes and vacuole does not contain DNA. Option 4 says nuclear envelope and mitochondria. I told you that mitochondria contains DNA, and nuclear envelope, that means they are referring to nucleus. That means nucleus also contains DNA. So the correct option for this is lysosome and vacuole. Okay. So Payal answered it correctly. Very good, Payal. Keep it up. Do this good work. Please keep doing this good work. Okay. Now moving next. Question number 3 says, which of the following cell organelles is responsible for extracting energy from carbohydrate to form ATP? Which of the following cell organelle is responsible for extracting energy from carbohydrate to form ATP? Option 1 says ribosome, option 2 says chloroplast, option 3 says mitochondria, option 4 says lysosome. Quickly answer me in the chat box, everyone. What you think is the correct option for this question? They're asking about extracting energy from carbohydrate to form ATP. Remember, I told you ATP. Everybody answer in the chat box. What you think is the correct option for this question? Which of the following cell organelle is responsible for extracting energy from carbohydrate to form protein? Pyle says C, mitochondria. Okay. What about the other students? One says 1. Okay. Or what you think? So as I'm running out of time, because we have to wind up this class at 1, so let me make it quickly. Pile is very good, beta. The correct option is C, that's mitochondria. Mitochondria, I told you, it also known as powerhouse of the cell. Remember, carbohydrate to form ATP, energy. Which of the following organ is responsible for extracting the energy from carbohydrate to form ATP? Remember the cellular respiration, that glycolysis which occur in the cytoplasm, from the glucose is break down into pyruvate, and then pyruvate move into mitochondria, where the pyruvate is converted into acetyl CoA. Remember, and then acetyl CoA, that acetyl CoA enter into the Krebs cycle. And the Krebs cycle, where it occurs in the mitochondria, the matrix of mitochondria. Remember? And then from that Krebs cycle, your NADH and FADH2 and your ATP and CO2 are formed, and then this NADH and FADH2, they move into ETS, which is your electron transport chain. Okay, and the ETS, your ATP synthesis will takes place. So Extracting energy from carbohydrate to form ATP is your mitochondria. But a ribosome does not play any role in formation of ATP. Ribosome main function is protein synthesis. Chloroplast, where chloroplast perform photosynthesis, and in photosynthesis, remember Calvin's cycle, right? Where there's a carbohydrate, which is very important for the formation of carbohydrate, right? And lysosome is it contains the digestive enzyme, like lipases. Okay, this lysosome, your chloroplast, and your ribosomes are not involved in extracting the energy from carbohydrate to form ATP. So the correct option is option number 3, which is your mitochondria. So quickly move to the next one, which says that non-membranous nucleoplasmic structure in nucleus are the site for active synthesis of dash. Non-membranous, remember, nucleoplasmic. It's a hint. Nucleoplasmic structure in the nucleus are the site for active synthesis of. Option 1 says protein synthesis. Option 2 says mRNA. Option 3 says rRNA. Option 4 says tRNA. Okay. The non-membranous nucleoplasmic structures in nucleus are the site for active synthesis of. Quickly answer me in the chat box what you think is the correct option for this question. Remember the hint, nucleoplasmic. Okay. Keepangrin, I hope I'm pronouncing your name correctly. Answer C. Okay, very good, Keepangrin. Option 3 is the correct answer, which says rRNA. Remember the non-membranous nucleoplasmic structure. That means they are talking about nucleolus. And I told you that nucleolus is the site where rRNA synthesis takes place. And nucleolus is also known as ribosome factory. All right, is known as ribosome factory. So protein synthesis is by ribosome. Or rough endoplasmic reticulum because it contains ribosomes. So protein synthesis, ribosome. mRNA, you'll see in transcription. Ribosomal RNA is the correct answer because the ribosomal rRNA is formed in the nucleolus. And nucleolus is also known as ribosome factory. Why? Because ribosome is made up of your rRNA plus protein, ribosomal protein. So it requires rRNA. tRNA is your adapter or a transfer RNA. So the correct option is option number 3, which is your rRNA. I hope this is clear to everyone. Quickly answer me in the chat box if it is clear. If you have any doubt regarding anything, please let me know. If any question or any topic is not clear to you, or if you want. Okay. I hope this session is informative for you. And you learned a lot of things. I hope that everything which I've covered so far is clear. Quickly answer me in the chat box because we are winding up the class. Okay. All right then. Please, student, try to do more previous year question paper, to more previous year questions so that your concept will become clear and you can have a more clarity about the exam and the topic as well. Okay. So thank you all. See you in the next class.